So let's talk about the transcription factors. So, where does transcription take place? Well, transcription takes place where the DNA is. And where's the DNA? Well, the DNA is inside the nucleus, in the nucleolus, right? And if you recall, right, transcription is basically where you're taking the information from DNA and putting it in a strand of mRNA. The mRNA will exit the nucleus via these pores, these circles in the nucleus, and will then go to the ribosome to be translated. So, if we zoom in, right, we'll be looking at the DNA, and let's look at a particular strand, just like one strand as a basis. So here's a strand of DNA, and so let's pause here. So here, we have this region called the TATA box. So the TATA is just a thymine-adenine sequence. So this is basically what we're going to call the promoter region. It's like the area before a start line, that where the pre-initiation complex for transcription is going to be made, so that we can start that we can start transcripting from the start point, which is this blue star right here. So, what enzymes are we going to use for this? Well, we're actually going to use 3 enzymes as the start of our pre-initiation complex. So we actually have TBP, or the TATA binding protein, and TF2A. And so what these proteins do is they're like magnets attracted to the TATA box. So they're actually going to guide both of them together, going to guide this third enzyme called TF2D, which is actually going to bind directly onto the DNA right before the start point for the transcription. And so this creates the base for our pre-initiation complex. And then, if you look here, we're also going to add this new enzyme, this TF2B, to the pre-initiation complex. And this really doesn't do anything important, so we don't need to worry too much about it. And this RNA polymerase, there's actually this green pinwheel, like a pin stuck into it looking thing, and that's an enzyme called TF2F. And that's actually going to act like the eye. It's going to be like, okay, where is this pre-initiation complex? So that it can go there and attach. And once it finds it, it's going to go attach to it. And now the engine for transcription is made. But we have to get the engine rolling. So we're going to use 2 enzymes called TF2E and TF2H to basically kick the engine into motion. So TF2E is only going to help kick it. The TF2H is going to help kick the engine into moving, but it's also going to do something else. It's going to cut the hydrogen bonds between the nitrogenous bases in the nucleotides of the DNA, allowing the RNA polymerase to go through and make a strand of mRNA without any struggles. And so, now that we've given it the kick, we can actually begin the transcription process. So now we're going to get rid of a bunch of enzymes, the motor proteins, the TF2B, and we're just going to leave the TBP and the TF2D at the TATA box. And now the RNA polymerase, with its eyes, the TF2F, is going to start moving along, transcripting. And so we actually have 2 strands to this DNA, right? So we have the template and the coding strand. So the template strand is the 3' to 5' strand, and this is the strand to which the RNA polymerase is actually creating the mRNA base complementary to. So this is acting as a template so that we can create our mRNA strand. And the coding strand, or the 5' to 3' strand, is going to be like rails. They're gonna guide the RNA polymerase and make sure it's staying on track to transcribe the template strand. And so this is going to continue. It's going to keep on going until it reads a terminator sequence. And once it reads a terminator sequence, the mRNA is going to tear itself off using a secondary structure. And now we have mRNA, but we aren't done yet. Now we're going to go to post-processing, where we're going to actually do a couple of things. One thing is that we're going to cut away these introns. So introns are just like junk pieces. They're like useless stuff that we don't need. So what we're going to do is we're going to have an enzyme called a spliceosome attached, and it's just going to go down. It's going to cut out these introns and glue together the exons. And once that's done, there's also going to be a modified guanosine cap added to the 5' end, and we're going to have a polyacyl or polyadenine tail added to the 3'. And now we have a finished chain of mRNA that is now going to go to be translated as the ribosome.